In this topic, we're going to discuss enzyme control and cofactors. So, by the end of this lesson, you'll be able to answer the questions: what is negative feedback? What is the allosteric site? What does phosphofructokinase do? And what are cofactors? So, what is a metabolic pathway? Well, a metabolic pathway is a series of biochemical reactions in which one substance is broken down or built up. Now, in a cell, you've got multiple metabolic pathways taking place. These pathways are very structured and tightly controlled. The reactions can be controlled by either varying the concentration of the substrate or enzyme, or the end product is used to control the reaction, which we'll have a look at in a moment. So what is end product inhibition? If you have a look at this reaction, you've got substrate A being converted into B, C, D, and finally you've got end product E. The different enzymes involved are 1, 2, 3, and 4. Now notice that end product E can move and bind to enzyme 1. When it binds to enzyme 1, it's going to be binding to another site other than the active site. It's going to be binding to an allosteric site. So it's another site called an allosteric site. When it does this, it's going to inhibit the enzyme, and so the substrate A won't be able to bind to the enzyme. If the end product is reduced, this will lessen the inhibition so that the metabolic levels will be brought back to normal. So this is called end product inhibition, and it's usually a form of homeostasis. So having a look at enzyme 1, because end product E can bind to enzyme 1, we say that enzyme 1 is a regulatory enzyme. It can control the rate of the reaction. So if enzyme 1 is inhibited, will substrate A be converted into product E? No. So enzyme 1 is a regulatory enzyme. And the way that product E and enzyme 1 bind, it's usually by non-competitive inhibition. So we say that enzyme 1 is a regulatory enzyme. Now, I've been discussing allosteric sites. What is an allosteric enzyme? These have got another site that can bind to another molecule, and the substances that bind to this allosteric site are called allosteric effectors. They can either be allosteric activators or allosteric inhibitors. When these allosteric effectors bind to the allosteric site, it's going to cause a change in the shape of the active site, and we say that it causes a conformational change. So allosteric activators change the active site so that it increases the affinity for the substrate. So the substrate will be able to bind. What do you think is going to happen when you have an allosteric inhibitor? It's going to change the active site so it can't bind to the substrate. So allostery means different shape, and an allosteric enzyme will change shape between active, which you can see B, and inactive, which is represented by A. In A, the allosteric site, when something binds to the allosteric site, the active site won't bind to the substrate. What about B? Is that enzyme active? Yes, it is. This is because the substrate can bind to the active site. Now let's look at what this means when we have inhibitors. If you've got an allosteric inhibitor, it's going to bind to the other side, another point on the enzyme, and this is going to cause a conformational change that the active site changes and the substrate cannot bind to the enzyme. As you can see, the substrate is represented by the little green blob. If you've got an allosteric activator, this is going to bind to the allosteric site, cause a conformational change so that the substrate can bind to the active site. Let's have a look at allosteric enzymes in terms of phosphofructokinase. This is an example of end product inhibition. Fructose 6-phosphate is a molecule that you find in the beginning of respiration. There's an enzyme called phosphofructokinase that converts fructose 1,6-phosphate into fructose 1,6-biphosphate. And how it does this is it uses ATP, so it uses a phosphate from ATP and converts it into fructose 1,6-biphosphate. This undergoes a series of reactions until you have got the end product ATP. Now, if the ATP levels are high, what ATP does is it's going to bind to the enzyme phosphofructokinase. When it binds to an allosteric site on the phosphofructokinase, it's going to cause a conformational change so that the enzyme is now inhibited. This means that the ATP levels will decrease. So you get inhibition of fructose 6-phosphate being converted into fructose 1,6 biphosphate. This means that the ATP levels will decrease. When the ATP levels are starting to decrease, the enzyme will be able to convert fructose 6-phosphate into fructose 1,6 biphosphate until the ATP levels have increased again. So this is an example of negative feedback. Now, what are cofactors? If you have a look here, you've got the blue representing the protein or the enzyme. A cofactor binds to the protein so that the substrates can also bind. So some enzymes require the help of another small molecule or ion to work. These are bound either permanently or temporarily to the enzyme. And they are non-protein components of enzymes that must be present for the enzyme to work. So let's have a look at cofactors. Now, cofactors can be either a prosthetic group, which is permanently bound to the enzyme, or a coenzyme, which is temporarily bound. So in terms of a prosthetic group, this is tightly bound, and the one that we know is the heme group, which contains iron in a ring structure. And you find this in hemoglobin. You've got some inorganic ions which mold the shape of the enzyme to increase the chance of reaction. For example, salivary amylase needs sodium to work more efficiently. And then you've also got some vitamins. So if you look here, you've got an enzyme, and when the enzyme binds to the cofactor, we call it a holoenzyme. So an apoenzyme is a catalytically inactive protein. So it's formed by the removal of a cofactor from the active enzyme. Your cofactor is a non-protein molecule or ion, and it's required by the enzyme for catalytic activity. So it can be an organic molecule or metal ion. So the coenzyme is usually an organic cofactor. So your apoenzyme plus cofactor forms an active enzyme. Here's some examples of coenzymes that you've probably come across. You've got NAD, and NAD is involved in the electron transfer chain. NADP, FAD, coenzyme A, coenzyme Q, thiamine, which is your vitamin B1, and peroxidine, which is vitamin B6, and vitamin B12. Examples of cofactors that you might have come across are zinc, iron, copper, potassium, magnesium. And zinc is used by the enzyme alcohol dehydrogenase, which breaks down alcohol. So what have we learned in this lesson? Well, we discussed end-product inhibition, and we looked at phosphofructokinase, how it's inhibited by the end product ATP. We also looked at allosteric sites, how when you have an activator binding to the allosteric site, it's going to cause the active site to bind to the substrate. When you have inhibition, the active site will change so that the substrate cannot bind. And then we also discussed cofactors, which are needed by enzymes to work properly. And that concludes our lesson. The end.